All right. So with that introduction, let's talk about this. I mean, like I just framed the name of the talk as a coronavirus as an untold story. So we're really going into the molecular detail of the replication, pathogenesis, antiviral vaccine development, and a small type of at the end, clinical retrospective research that I actually do. So with that brief introduction, let me start with the material. So this is the outline of the talk. We're going to talk about the classification, evolution, and then structure, going into the infectious cycle. We're going to talk about the entry into the host cell. And then we drill down into some of the molecular ideas about the transcription replication and where it happens, and touch on pathogenesis immune interaction, and also going to cover the antiviral and vaccine development, and finish with a small retrospective study that I just do in collaboration with other people. So a lot of information, but just bear with me in that, because it is almost impossible to keep updated with even the latest information and the research, because every day hundreds of literatures are coming out. So that's what the issue is, and you have to pick and choose that what you can present and what your audience is. So being said that, the first thing is that where this belong. So the coronavirus actually do belong to this coronavirus family, which is under this nidovirales, which does have a specific type of nested genome RNA to mRNA synthesis, subgenomic. We're going to talk about that. And then they are mainly the alpha, beta, delta, and gamma, but the alpha and the beta are the only one which infect, these are the coronaviruses which infect the human beings. Now, some of these, particularly the A229E, which is a human coronavirus, 229E and the OC43, these two were known for a long, long time. And after the SARS outbreak, the NL63, HCOV, HKU1, these were identified much later. But all of these are actually much, much milder kind of coronaviruses, which are responsible about 10 to 15% of the common cold that we see. Now you can easily see that right here in the beta coronavirus, the SARS of 2002, 2003, that epidemic that's stopped, that also is there. Then a MERS coronavirus, which is a Middle Eastern Respiratory Syndrome coronavirus, actually came out or first reported in 2012, much more lethal. And we're going to talk about that. And now the current SARS-CoV-2, these are all under the beta coronaviruses. So the next thing that what we are going to talk about is how these coronaviruses actually evolved. So most of the coronaviruses actually evolved from some unknown ancestors, and then they were the reservoir where the bats, different type of bats, horseshoe bats and many different types. And all the alpha and the beta coronaviruses, the bat is usually the ancestor that what we see, and the transmission is usually zoonotic. What we mean by a zoonotic, that it jumps from the bat to some intermediate host and from some intermediate host to the human beings. That is normally seen, but we do not know that whether from bat it can directly jump into the human population as well, that we have probably guessed, say for example, in Ebola, Nipah virus as well. So on the other hand, the gamma and the delta coronaviruses, we do not have any human species or genus of these viruses, and the birds or the avian population is generally considered as the ancestor. So being said that, if you just look at the lineages of the coronavirus under the alpha, beta, gamma, and the delta coronavirus, I'm just looking at the human coronaviruses, we have these 4 lineages, and these are based on the genomic sequences, and you can see that they are grouped in lineage A, B, and C, and D. And the SARS coronavirus 2 actually evolved as a B, and it's spreading out into A and C as well. So it evolves. And these are small changes in the nucleic acid that you can easily see. So just this classification is based on one key replication protein, which is just the RNA-dependent RNA polymerase. And you can just do that as a genome-wide scanning of this material. And we do have very strong proof that this is actually not man-made, because we do have some particular justification for that, because it is close to a bat coronavirus or a TG13. but not any of the coronaviruses which is close to the human being. So it is very unlikely that just a virus which is 96% similar to a bat coronavirus, just a couple of substitutions that you start with and it's leaked out from a lab. And again, there are many other, because beyond the scope of this talk that we can talk about, we know that it is non-man-made from the genomic sequences, because there is a molecular signature in these viruses which tells us that it is not man-made. So just an overview of that the zoonotic reservoir is bats. Probably more than 500 different genus of coronaviruses are known, and these are hugely undersampled. So there could be more than 1,000, 5,000 of them actually can exist. So we know the intermediate host for the classic SARS is a palm civet cats, whereas for the MERS, it is the dromedary camels. These are the Arabian camels or the Saharan camels that we actually see with a single hump in their back. And SARS-CoV-2, or we say that often as a SARS-CoV-2, that pangolin is being suggested because there is a certain amount of sequence homology with isolate from the pangolin, but we really do not know, because there is no situation that actually tells us that it came from the pangolin. It is just been suggested, but there is a lot of it's completely speculation still yet. That palm civet dromedary camels, we have actually seen them. That's the virus that is transmitted to the human being. But from the pangolin, they're similar, but not the same. So you can easily see that for the SARS, it was about this much of cases, about 8,000 cases and 774 death. Whereas the Middle Eastern coronaviruses actually cause almost 30% death. And this is by far the deadliest of the coronaviruses. And there is still sporadic cases that are coming out, but it is not in a status of a pandemic or an epidemic. SARS, the epidemic actually ended in 2003. So as you can see that if you just look at the COVID-19 as of now, if we just look at this particular data from now, can you see my screen as such? Let's see.
Yeah.
Can you see my screen? Okay, so this is the live basically the numbers that you can see, that is 40 million global cases, 1.11 global deaths. So you can easily imagine that there is no end in sight at this point, that how many will be infected whatsoever. So that's where it stands. So let's go ahead with the rest of the material, which will be the much more detail about the molecular virology type of material. So first, we are looking at the coronavirus structure. These are the viruses which are about 100, 150 nanometer in diameter and does have a 30 kb huge RNA genome. And it's a plus RNA genome, which means that it is a 5' to 3' genome, which is positive sense, means it's ribosome ready as soon as it gets into the cell. And the envelope that you actually see right here is covered with a spike protein that you all know. And then there is also another protein, which is also a transmembrane domain with a transmembrane domain. All of them is the membrane glycoprotein and matrix protein. And you can see that it tethers the genome with this envelope of the viral membrane. Now, this genome itself is an RNA, but it is not in a naked form. It is covered by a protein which is known as a nucleocapsid protein, and together it makes a ribonucleoprotein complex. And then it is covered by an envelope. This is a phospholipid bilayer, and these phospholipid bilayer is derived from the cell host membrane in which these viral proteins are inserted. And this is one of the reasons that if you just can think about that, why hand washing just with soap and water is effective enough, because the detergent molecule actually just get inserted into this lipid membrane. And if this lipid bilayer actually get destroyed, then the virus is inactivated immediately. And this is something you can think that this is lipid, so that this is like your oil and grease, and you use soap to wash your hands to take them off, and the detergent or the soap does the same thing for the membrane. So huge genome, and we're going to talk about that. and one of the largest genome out of all the different RNA viruses. There is a idea that you can maintain a certain size of the RNA genome. After that, it becomes really unstable, and you have to go into the DNA genome. And these are some of the largest one, and we're going to talk about that, how they can maintain it. So again, genome is a plus-sense RNA encapsulated with nucleocapsid protein. It is a 5' capped with a poly A tail. That means the genome is ready for the ribosome to be translated. And all of these RNA viruses code for an enzyme that is RNA-dependent RNA polymerases. Now, just think about for a minute that as this is ribosome ready, the virus doesn't have to package the intact RNA-dependent RNA polymerase enzyme into the virus, because this can be translated into the protein, which can make the RNA-dependent RNA polymerase, which can actually make more of these RNAs by transcription and replication. Whereas if this is a negative strand RNA virus, say for an example, vesicular stomatitis virus, or say Ebola virus is a negative strand RNA virus, that is not ribosome ready, and the virus actually packs its RDRP, or the RNA-dependent RNA polymerase, in the virus. And as it comes into the cell, it has to make a plus sense RNA from that minus sense RNA so that it can synthesize its protein and subsequent other reactions. So this is this code for the RDRP, but it doesn't package into the virus. So this is how the nucleocapsid protein enwraps this naked RNA. So this is basically a single cell overview of this. And we're going to see that coronavirus enters the cell. The genome RNA is translated immediately into some polyproteins. So we're going to talk about that. And then there are two processes, the transcription and the replication processes. And after that, it is assembled into this endoplasmic reticulum Golgi intermediate complex to form the smooth wall vesicle and then egress from the cell. So now we are going to talk about these spike proteins, and basically this is the part which is the entry, that how it actually happens. So this spike protein is called a classic type of fusion protein, and it's a class 1 fusion protein, which is made up of alpha helices, and it's a trimer. There are 3 monomers in there. You can see 1, 2, and 3. And in this structure, what you see that there is a globular head, which is the S1, subunit 1, and then S2. Each of them has the same thing, which is the stalk. And then these 2 parts are separated by a linker, and you can see that in this linker, there is a unique sequence, which is this polybasic site. You can easily see that you have NSPRRA. This is a site that we have not seen in the regular or the other SARS or the MERS viruses that is in there. And we're going to talk about that, what it actually does. Now, here you can see that there is a fusion peptide, FP, and this fusion peptide is responsible for the fusion of the viral membrane to the cell membrane, and this must be exposed by a proteolytic cleavage that we are going to see. And this is the receptor binding domain of the virus, which you all know that it binds to the ACE2 receptor on the cell surface. And these are some of the heptide repeat, and these are some of the transmembrane regions of that. And let's take a look at the rest of the structure in a second. So if you just look at the spike protein in general, what you are seeing that all these, the purple colored that structure that you're seeing, these residues are conserved, whereas the ones which is in this aqua color, which is in a variable region. So this is the side view, and you are looking at the top view in here, and you can see the monomers, one monomer here, another here. And this actually makes it a threefold symmetry of axis right here with 1, 2, and 3 monomers in each. So you're seeing this is the top view. And right here, what you are going to see that right here, you can see there is a purple residue right here. And this is a conserved residue. And this is a unique residue change of a couple of amino acids, just 5 amino acids in the SARS-CoV-2, and extremely conserved. And just these 5 amino acid changes actually allows this binding to be almost 20% more affinity than the classic SARS-2. Classic SARS-2 also binds to the same receptor ACE2, but not MERS. MERS has used a different receptor, which is called a DPP4, dipeptidyl peptidase 4. But SARS classic plus SARS and this use the same virus or the coronavirus 2 use the same receptor ACE2 to enter. And this just changed this sequence, actually make this virus bind more tightly, and that is attributed to the high infectious rate of this virus. So if we look at the virus, that how it enters, there are a series of protease cleavage are needed for this virus to be even released. And before release, you need a protease cleavage of these proteins. And then before the attachment, before the virus actually enters, also you need a separate, different type of protease cleavages. And we're going to talk about that what it is. So if you look at the top, that this is a linear sequence of the spike protein, and here is the fusion peptide right here, and this is the internal fusion peptide. And before that, you have an S1, S2 protease cleavage site, and also another S2 prime cleavage site. So the first cleavage that needs to be done is cleaving that area right here in between S1 and S2 somewhere, either both of them or just one, to expose this fusion peptide. Then a second protease cleavage cleaves right here in this site to expose these internal fusion peptide, which buries into the membrane. And what you are seeing that this 2019 nCoV, this is before we named this publication came out before it was named as the SARS-CoV-2. So this is a 2019 nCoV-2 is the same thing, novel coronavirus. And you can see right here is this polybasic sequence, which is not present in the SARS-CoV-2 and the others. So this is a unique site that has been introduced in here. And this is actually a site which can be digested by a furin-like protease. And this is an extra site. And we think that this has something to do with the virulence. So chances of a protease digestion are increased to 2 of these, and that may offer some extra virulence in the process of the entry. So that's basically what it is. So let's take a look at how it happens and where this. So first, the spike protein binds to the ACE receptor, and it is digested by this TMPRSS2, which is transmembrane protein protease, serine-2, and removes this receptor binding domain away from that and exposes that fusion peptide, which is inserted into the membrane, and the virus gets in. So how it happens that you can see that at the top is basically the cell membrane, double membrane, bottom part is a viral membrane, and this receptor binding domain is still there, and this receptor binding domain is removed by the protease cleavage, which underlying this red things you cannot see right here, because these are the fusion peptide, which are hydrophobic amino acid sequence, and that's why they are tucked inside the protein. And due to the protease cleavage, these are exposed and inserted into the plasma membrane of the host cell. Now what happens, and a remarkable transformational change, conformational change goes on. And in that process, these 3 alpha helices at the N terminal and the C terminal actually bundle up into a 6 helix bundle. And these bundle actually brings the top and the bottom membrane together, and then forming a fusion pore through which the viral nucleic acid or the payload can be delivered into the cell. So here is a animation of that, how it happens. You can see that the top is the virion and the bottom is the plasma membrane. And this is right here is the protein. And we are going to just see that how the first ACE2 binding and a protease cleavage, which is not shown, will take out the receptor binding domain, which is take out this, and the fusion protein, fusion peptide, which is inside, is exposed and fall onto the membrane, and how the fusion actually occurs. So let's take a look at this. Glycans are the carbohydrate residues. Removal of that's the fusion peptide inserted into the membrane. And this is another one, 2 of these. And this is a hairpin formation into a 6-helix bundle, which brings that viral membrane. And now the cell membrane is continuous and the virus can deliver its payload. All right, that covers pretty much the entry. Let's go into the transcription of the genome replication. So genome is a large genome, about 30 kb, and does contain a replicase gene, which is coded by this rep1a and 1b. We are going to see that. And this code for 16 different proteins, including the RNA-dependent RNA polymerase. Whereas the 3' end of the genome actually produce this structural and accessory protein. And out of those, the main one are the spike protein, the envelope protein, the membrane protein. These are all on the cell surface, on the virion surface, on the membrane. And this N is the nucleocapsid protein, which enwraps the RNA. And then there are some other proteins like hemagglutinin esterase that is not found in the SARS-CoV-2. They're found somewhere else. And then these ABs are actually accessory proteins which help in the packaging and other function in the virion. So you can easily see that this is a mouse hepatitis virus. This is a classic SARS. This is a feline cat coronavirus. This is an infectious bronchitis virus. This is an avian coronavirus. And there is substantial difference in their genome organization. So how these are organized, we are going to talk about in a second. So if you can see that this 30,000 base pair genome, these are the open reading frames, but there are just only 10 of them. But the virus actually codes 27 different proteins, and how it does that from these number of ORFs, because you would just expect that as per eukaryotic translation, that 10 ORFs, 10 proteins. But it is not so, and we are going to see that how this actually happens. It's a remarkable process. So the first thing what happens that when the virus enters into the cell, this is the genome that comes into the virus, and these are the different open reading frames. The first thing what happens that it is translated by the cellular ribosome into a protein, which is a 1A protein, and then by a ribosomal frame shifting, which is a controlled program process, it produces an 1A and a 1B protein fused together. And these are called the polyproteins, which are made up of many proteins, including some proteases. These are the viral proteases. Do not confuse that with the cellular proteases, which actually helped in the entry. These viral proteases cleave them into 16 different proteins, including the RNA polymerases holoenzyme. And after that, what happens is a remarkable thing, that the first thing in that process is that this middle thing is the genome, which is the plus sense RNA. From there, the virus actually synthesizes these subgenomic RNAs. And we are going to see that it happens in a remarkable way that actually happens, that this is the plus sense RNA. From that, it first synthesizes a partial minus sense, and that is the subgenomic RNA. But what you are going to see that it starts, the RNA-dependent RNA polymerase starts the transcription from this end, and then it reaches a transcriptional regulatory sequence from where it jumps and then finish at the other end. So in this process, what happens, all these subgenomic mRNAs will have an exact same 3' end, also the same 5' end. So 3' end and the 5' end are the same for all of them. And what you can see that most of these are the polycistronic mRNAs, except the last one. And these are the mRNAs which translate into the structural proteins, but only the first gene in this or the first ORF open reading frame is translated. So from this, only 2A will be translated. From here, hemagglutinin esterase will be translated. From here, only the S will be translated. Rest will not be translated. And that's how the mRNAs are translated into the structural protein. Then the next part, what happens, that the full length of the genome is also synthesized by this minus intermediate, where the polymerase doesn't jump from these sequences, which are the transcriptional regulatory sequences. The ribosome, the transcription, the RDRP reads through the whole of the plus sense all the way to the end. And this is the intermediate minus strand on which it builds the plus strand genome again. So it starts with the plus, makes the minus intermediate, gets back the plus. For the transcription, same. You just make the minus complementary sense from this plus sense, and you get the plus subgenomic mRNA, which is ribosome ready. So the olive ones are right here are the minus sense. And then you can get additional translation from that to which is basically the same. So let's take a look. It's a complicated picture that how these polyproteins are synthesized, that in between the junction of the rep1a, 1b, there is a sequence which we call as a slippery sequence, this UUA-AAAC, followed by what is called a RNA pseudoknot. It's an extensive secondary structure. And in this position, the ribosome actually slips over this UUA-AAC sequence. And this is enhanced by this RNA pseudoknot and sometimes slips the ribosome out of the frame, just -1. If that happens, what happens? That there is a stop codon right here, UAA, at the end of the this replicase 1A gene, and this is not recognized anymore, and the ribosome reads through this stop codon and synthesizes that rep1b. So 20% of the time this happens. So it synthesizes the rep1a most of the time, and in some cases by the ribosomal frame shifting, it doesn't recognize that and synthesize the rep1b. And in this process, it produces 16 different proteins. So from the first, the polyprotein A is this, and you can easily see that these are the proteins. And again, you do not have to go into the in between each of them in detail, but these are the proteins which are synthesized. And out of them, there are a couple of them. You can see that NSP1 is an interferon antagonist in here. And there are some 4 and 6 are the transmembrane scaffolding protein, whereas the NSP5 is a main protease that we talked about, which will cleave these protein at these sites, which will make these individual proteins from this long polyprotein. And then the second reading frame will synthesize the rest of the protein. Out of that, this NSP12 is the RNA-dependent RNA polymerase, which in association with NSP7 and 8 form, along with the others, form the RNA-dependent RNA polymerase holoenzyme. And just note of just one of these, NSP14, this is a 3' 5' exonuclease. And this is a remarkable enzyme that we find only in the Nidoviralis with this such a long genome. This does have a proofreading activity unheard of in any of the RNA viruses. And this is one of the reasons that the nucleoside analogs like ribavirin, even remdesivir, favipiravir, this actually doesn't work because it is excised out by these proofreading enzymes. So you can easily see that how this actually works, that making from 2 ORFs, you can make 16 of these polyproteins. So the next part that we are going to see that you can easily see that the amount of the proteins that is produced is controlled by this molar amount in a fantastic way. And easily you can see that the control of the transcription and the replication, just the core holoenzymes are these, and the accessory RNA protein, they're all made in a respective amount just by this ribosomal frame shifting controlled by the pseudoknot. So how this actually happens, you can see that these are the extensive the extensive stem loop structure RNA secondary structures, and you have these sequences at the 3' end, and also you at the 5' end you have these TRSL, which is called the TRS leader, and then in between these genes you have these TRS or the transcriptional regulatory sequence body. And we're going to see that how the RNA polymerase actually jumps from these sequences to the leader. And these are some of the conservations that you can see in between them. So what happens that suppose the RNA is being, the minus strand is being synthesized from the 5' end. So this is the template plus strand genome RNA, and it starts synthesizing the minus strand from here. And now it comes to this core sequence of the translational regulatory sequence, core conserved sequence. And this sequence is similar to a sequence in the TRSL, which is all the way down, 30,000 base pair is right here, all the way down. And this can actually jump over the period of many of the time to go into the other end this way and switch the templates. So this is how it can jump from one place to the other. So this is just gives you an overview of the holoenzyme structure, that so many different proteins are responsible for the replication process. So here we are looking at some of the subsampling of the, let's take a look, of this, that how this actually evolved. You can see that it evolved as this blue, which is a beta lineage, and then just moves into some of the other lineages. And these are all based on the whole of the sequence. And if you can see that there are some mutational hotspot in somewhere, but what we actually see that right here, where it is a spike protein right here, there is a mutational hotspot, but it's not mutating too much as such. And we believe a vaccine will work for that reason. So let's go into the next material. All right. So next we are looking at the replication transcription complexes, that where it actually happens. So many of the viruses produce that, and these are the double membranes. So you have two membranes, and these membranes are set up inside the cytoplasm. And here the viral replication actually happens. And there are some of the reasons for it does that: escape from the cell defense, and also concentrate the transcription replication components into these. And when we have seen in an experimental process that what are the proteins present in there, you can see that there are lots of these proteins which are related to cellular EIF3, EIF3F. These are cellular translational factors which are present in there. So what are the microenvironment in these replication transcription complexes? You can easily see that all of them in the red, these are the 16 proteins which are all replicase gene proteins. They are in a higher amount, whereas you can see that NSP1, which is an interferon antagonist, that is less, whereas the other structural proteins are less, except the nucleocapsid protein, which enwraps the RNA. So you can easily see that how the replication actually happens in these RDC from these experiments. So this is also another, that how it has been shown that active translation happens near the active RTCs. And you can easily see that puromycin is a translational inhibitor, and it inhibits that. But when you merge without any treatment, you can see that the active translation actually happen in very close. The NSP8 is one of the protein which is already present in the RTC that we have seen in the previous one. NSP8 is a large amount, and we are looking at the double-stranded RNA which are present, and it happens very actively close to the translational sites. So how it is organized? This is a very recent publication in the Science, that you can see that these are the double membrane structure from the electron tomography. These are the ribosomes, and you can see in this structure, this is an intermediate Golgi endoplasmic reticulum Golgi complex, where you can see the spike proteins, the ribonucleoprotein components, and the virus is getting assembled into this structure. So the overview of that is that this is also another pore complex that is made by these NSP3 protein, which is set up on these membrane vesicles, double membrane vesicles, which allows the viral RNA to move out of these replication transcription complexes into the cytoplasm, either for packaging or for translation. So this is just a packaging model, that right here is the NSP3 on the membrane, and these RNAs are coming out and immediately encapsulated by the N protein, and then these proteins will be associated with the other structural protein right here, including the spike protein, the envelope protein, and the membrane protein, to form the viral particle as such, which will egress from the cell. So that is the overall process. So that's basically, we talked about that once these are done, that these are the endoplasmic reticulum Golgi intermediate complex, where it derives the membrane. And once it derives the membrane, all these viral proteins are inserted into that cellular membrane derived viral membrane, and the virus actually is expelled out just by the process of exocytosis. So here we are looking at some of the pathogenesis and immune interaction, that how you evade the host defense immune system in this process. that some of them actually are able to do this, but very few of them, but that is good enough for an infection. So what we are looking at, that coronaviruses actually can be detected by many of this, and these are mainly the TLRs, or the toll-like receptor, which can which can sense the double-stranded or the single-stranded RNA. But what happens that when you concentrate all or most of these RNAs into those RTCs, or the replication transcription complexes, that evades this cellular responses. And that's one of the reasons that it can escape escape from these inflammatory cytokines in the initial part. So that's one of the reasons that it actually happened. And this is silenced by this NSP1 as well. So several ways that coronaviruses can evade the early detection of the viral RNA. So how this NSP1 actually does that, you can easily see that NSP1 binds to the ribosome, and it doesn't allow the host mRNA to be translated. So if there are interferon-induced genes which are antiviral, it will suppress the host translation. But this binds to a 5' untranslated region of the viral mRNA, and it allows the virus to translate them preferentially. It shuts down the host, but it allows only the viral mRNA to be translated into the proteins. Very, very clever method by NSP1. And this structure has been solved as well, that how NSP1 actually blocks the 40S ribosomal subunit in a little bit of more detail. All right, so after that, we are looking at that what happens, that you can see that early interferon signaling, there will be much more milder disease. And if there's no interferon signaling in this process, also our immune system have other processes by which we can minimize the disease. But in the delayed process, what we see in the SARS, you can see that there is an exponential growth of this cytokine storm, which actually cause the pathology. And again, we do not have to go into the detail, but if you just look at that, the first one is the healthy control, and then the ICU and the non-ICU patient and the level of their different cytokines. And just to have an idea, you can see that the green ones are the pro-inflammatory cytokines, and the red ones are the inflammatory cytokines and the chemokines. And all of these are elevated by this viral reprogramming of the immune function, which is the delayed IFN response. And that actually caused the pathology, what you are going to see, that this is an alveoli in the lung, which becomes fluid-filled due to this. Lots of proteins are produced in the later amount, and that actually helps in the building up of these scar tissues, hyaline glassy membrane, and complete surfactant removal from this, and the gaseous exchange almost becomes impossible. So here you can see that these are the pneumocytes and which can get infected, and this is the surfactant inside which is needed for the gaseous exchange. And in an acute infection and the inflammation, due to the debris and the other proteinaceous material, these surfactants are actually completely being diminished, and also the pulmonary edema or the water accumulation or the fluid accumulation, which stops the gaseous exchange in between. So that's basically the pathology. The next part that we are going to talk about quickly, that the antiviral and the vaccine development, that how it actually happened, that we start with this, and over this period of time, which takes 10, 15 years, many of these drugs are rejected. So we are not starting any drugs as such, but we're trying to repurpose some of the drugs which are actually available in the market. So where we stand. So you can easily see some of them, that what are the different things, and I'm just touching a couple of them. Say, for example, one of them is a spike protein target, and it's a monoclonal antibody cocktail. I just say that is a Trump device from Regeneron that is in the phase 3 clinical trial. And you can see that endosome fusion target hydroxychloroquine, we have used this Epinavir. This does not work because there are other processes. Remdesivir is an RDRP target, but remember that all of these nucleoside analogs are excised out by the exo-N NSP14, and that's why you can see that the WHO actually just announced the day before yesterday that remdesivir actually doesn't help in this process. Also, viral protease inhibitor, which are the lopinavir, ritonavir. This is used in the hepatitis C virus that is being used. not with much success at all. So still, it's a long way to go as such. So here it shows in vitro that remdesivir can actually work. And you can see that this is a science paper. So everybody was jumped into remdesivir seeing that, okay, all right, here is an elongated RNA and remdesivir is actually completely halting this process. Yes, it happens in the test tube. Very good. But it does not happen in vivo in the body. So this actually is a structure of the remdesivir, which is bound to the RNA-dependent RNA polymerase in the active site. You can easily see that. Right here is the nucleoside analog, great for the publication. It does work, but in vivo, the enzyme excises it out. These are just a couple of different in a list of different drugs that is in there. These are the IL-6 or the interleukin-6 receptor monoclonal antibodies, which actually block them. And there are many in development, and I'm not going into the detail of that, but most of them are the immune system target as such. There are a couple of strategies of the stem cell approach, which is also for regeneration of the lung tissue. These are the companies which are doing that. So the next quickly part we will just do is the vaccine development, how it is happening. So the different types is that the first one is an attenuated vaccine where you take the virus, passage them constantly, and sometimes it becomes avirulent, but actually retain its antigenicity to be used in the vaccine development. Another is an inactivation by the formaldehyde or heat. And these are some of the classic methods of vaccine development. In addition to those, what happens, you can see that you can make recombinant subunits, which are the protein subunits. You can use that. Also, in addition to that, you can use the cloning method to use an mRNA vaccine. You can make a DNA vaccine. You can make expression of subunit proteins and also make virus vector vaccines. So we are going to just look at a couple of them. So here it's a busy slide, but just where we stand right now. So if we just look at that the first one, which is a very famous mRNA 1273 Moderna, that is made up of these mRNA, full-length mRNA of the spike protein, and Pfizer is and BioNTech making this BNT162, which is a part of this spike protein mRNA. On the other hand, you can see that the yellow ones are what is going on in India. The DNA vaccine, which is a Zydus Cadila, is being making, whereas this CoronaVac and the Covaxin, these are the inactivated whole kill virus, and this is the classical method. Whereas this mRNA and this adenovirus or the VSV-based, these are the new methods. And also the Novavax also using a nanoparticle and delivering some of the spike-like proteins into the cell as well. So we are going to talk about this, how these two methods actually work in a very, very quick way. So the first thing is that first, the amplifying RNA vaccine mechanism, you can just clone the RNA that is needed into a DNA plasmid and also put a replicase which will replicate that RNA and deliver that. When you put this one in the cell, you can easily see this replicase will make these transgene into these mRNAs, and these mRNAs code for the proteins which will be translated by the ribosome, and these will actually induce the antibodies in there. On the other hand, you can package them in a virus-like particles and deliver them, and it will do the same function, just different delivery method. On the other hand, you can see that there is a direct method that you can just take the replicase and the transgene, mix it with the saline or the adjuvant, directly inject into the cell, which will make from the positive sense into a negative sense mRNA, and the replicase will make the mRNA transgene. Now, these two methods are actually now being used where you are seeing self-amplifying, trans-amplifying RNA, where you have two of these going into the cell separately. So you have a mixture, not together in a cis, but in trans, separate two material. You add the replicase as well as the transgene, and this replicase actually make into the mRNAs and translate it into the proteins, and it makes a large number of these mRNA transgene, which is making the vaccine antigen. Whereas we have seen a non-self-amplifying replicase even enhance this amount of mRNA transgene production because you are not giving this replicase, and the cell is actually much more comfortable rather than making 2 copies of these foreign proteins, just 1 copy, and the enhanced immunogenicity that has been suggested. So the next thing that I'm going to talk about, that how the adenovirus vectors are made. You can easily see that the adenovirus gene, the whole genome is taken, and there is a protein called E1, which is dispensable. It's a transcription factor. You can cut this out by homologous recombination, and in that you can just insert the protein of your interest, which is the spike protein. And this is a recombinant adenoviral vector, which you can package into the virus, and this you can deliver into the cell. And that will basically deliver this spike protein gene to the cell to be produced. Also, another thing, you can directly do this by the cloning, same process, but just rather than a homologous recombination, you can just cut them with the restriction enzyme and package them into the molecular plasmid, which you can make into the cell, into this viral particle, which you can develop. So the Oxford vaccine is using a monkey chimpanzee adenovirus, whereas the adenovirus 5, which is a human adenovirus, which is attenuated, is being used by the other companies at this point. So the last couple of minutes, I'm just talking about that what we were talking about, this retrospective clinical study, and how this type of research work, that we collaborate with the hospital and the clinicians and collect the data, and some way we actually report these to the journal. And unfortunately, it is not a very popular work, this type of work. So how this actually, we have worked with this, that say for an example, that this came out just 2 days back, and now all these hundreds of publications which actually was proposing about the remdesivir just have a scientific paper value. It will not be in the clinic, and that's the reason that this type of research is in there, just to show the efficacy of anything that we jump onto them and try to find the magic bullet. So in this process, what the doctors have used, that we collaborate with, they took interferon alpha, which is a known antiviral agent, by making many of these antiviral products. You can easily see that the hyphen defined the boundary of the tissue, and this is how it actually works. Not going into the detail of that, but we use this. The doctors use that interferon alpha 2b. They also use this lopinavir, and you can see that this is an HCV protease inhibitor. And if you remember the genome of the coronavirus, these are the sites which are digested by this papain-like protease and this 3CL protease, and they assume that this will be also inhibited by this protease inhibitor. So this is the second one. And the third one is that umfenovir, which is also known as Arbidol, that is used in Russia as well as in China as a drug for influenza. It is not authorized for use in United States, and it helps in the viral entry, similar to hydroxychloroquine. And you can see that. So these are the triple combination studies that has been given to the patients at the early stage of their admission. And you can easily see that most of these patients, you can see that the symptom to admission is about within 3 days. They were admitted to the hospital, and their average length of stay was about 16 days. And from admission to negative PCR result was about 12 days. So this is about the study that we have seen. And then if you just look at the correlation, you can see that the age does have a good correlation with the age. You can see the p-value. That means that if you're aged, you will probably be in the hospital for a longer period of time. But there is no gender. And when we adjusted it with the age, gender, and the imported cases from other area, the length of the hospital stay or the admission, length of the stay, as well as the negative PCR results, we did not find, if you just look at these values, we did not find any of these as meaningful. That means that even these combination studies, it is actually in clinical setting, it is not working. And unfortunately, many of these studies initially came out that say touting that they are actually working symptomatic studies, but unfortunately, they are not. So I will stop there, and I can take some questions as well. Sorry for going over a little bit. And these are my collaborators and acknowledgement.